Hello friends, welcome back to my YouTube channel. Today in this video we'll be going to see the topic of 101, that is yeast 2 hybrid assay. Okay, so what is this? This is the protein-protein interaction. Okay, it is a type of a protein-protein interaction. Protein-protein interaction can be done by two ways, that is yeast 2 hybrid assay and another is phage display. Okay, we'll be going to see the phage display in a meanwhile time, but before that we'll be going to see about the yeast 2 hybrid assay. Okay, so what actually this is about? Okay, it has actually, it depends on, it is based on the transcriptional factor. transcriptional factor Okay, it is based on the transcriptional factor and activation of transcriptional factor, how it is going to activate. Okay, so yeast to hybrid assay has two domains. It has two domains. One is DBD domain. Okay, DBD is nothing but DNA binding domain. Okay, so it is called as GAL4 DBD domain. Okay, and other domain is, second domain is A D domain. A is for activation domain. Okay, which we can say as GAL4 AD. Okay, what these domains do? These domains are joined with the proteins. Okay, they are joined with the proteins, and these protein interacts with each other in this system. Okay, these proteins are interact with each other in this system. So what are these two proteins interacting? What are their names? Okay, for gal AD, prey is the protein, and for gal DBD, bait is the protein used here. Okay, so these two protein interacts with each other. When they interacts with each other, they will regulate the transcription. Okay, when they interacts with each other, they will regulate the transcription. If they don't interact with each other, what happens? They will off the transcription. Okay, so actually when they interact with each other, they will become a functional transcription factor, and then they will regulate the transcription, and we can identify the reporter genes. Okay, if they don't interact with each other, we cannot able to identify the reporter genes, and the transcription will turned off. Okay, so we'll be going to see this with the help of a diagram. See, this is the diagram. Here you can able to see this is the DNA binding domain, and the bait is attached to it, where bait is interacting with it, and the prey is interacting with the activation domain, and activation domain is attached with the RNA polymerase. Okay, and when this whole process occurs, transcription will turn on, then the transcription will goes on. Okay, now we'll be going to see how it is going to work. GAL4 AD, I said you what is AD, activation domain, and this is the prey protein which is attached with this GAL4 AD, and this is a bait protein which is attached with GAL4 DBD. DBD is DNA binding protein, and when they both interacts with each other, they will form like this. And after that, these two proteins interact with each other, and then the functional transcription factor will forms like this. Can you see this, what I have wrote? Functional transcription factor. See how the transcription will turn on and how it will turn off. When upstream activating system occurs, here upstream promoter, downstream will be there in the transcription, right? We know that, we have studied about the transcription, where upstream will be there, downstream will be there, -10, -20, +10, like that, right? You know that. So what happens here? Gal for DBD and gal for AD will comes and when the protein binds with each other, they will come together and they will turn on the transcription. And when they turn on the transcription, we can able to identify the reporter genes that can be a fluorescent or lag Z genes. We will be going to study about the reporter genes in a separate topic, because you are having a separate topic of this reporter gene. So when the transcription turns on, transcription will goes on. Now if the protein doesn't bind with each other, no interaction takes place, then what happens? Transcription will turn off. Okay, transcription will turn off. So this is the whole process about yeast to hybrid assay. Now let me show you the notes how I prepared, so that you can able to join in my batch. Guys, you people were telling me that why I'm charging for the notes. See guys, why I'm charging is because I'm doing the hard work. See here, I'm providing you the simple and beautiful notes. Here I have write first point I wrote about the protein interaction. Second point I told about the two distinct domains. Then what are those I have wrote here, and then I have written about the then I have written about about the system. Like we will introduce these proteins in the yeast cell. Which yeast cell? The yeast cell which will lack these proteins. Then we will clone those proteins in that yeast cell. So I have wrote about the whole detail about the bait, how bait is going to attract and how prey is going to interact, and how we are cloning this. Everything I have wrote in the detail along with the diagram. Hope you loved this notes, and you will join in my batch. If you are new on my channel, please subscribe my channel, share my channel with your friends. Thanks for watching.